Hello and welcome back to the channel. Today, we're diving into a fascinating area of cancer research that doesn't often make headlines, but has generated significant interest in scientific circles and online communities alike: the potential anti-cancer properties of certain antiparasitic medications. Specifically, we'll be examining Ivermectin and Fenbendazole, two well-established antiparasitic drugs that researchers are investigating for possible roles in cancer treatment. But before we begin, I want to emphasize something important. This video presents current research and is for informational purposes only. Nothing in this video should be taken as medical advice. If you're dealing with cancer, please work with qualified medical professionals on evidence-based treatment plans. With that said, let's explore what the science actually tells us about these medications and cancer. Separating fact from fiction, laboratory findings from clinical evidence, and promising leads from proven treatments. Drug repurposing, finding new uses for existing medications, has become an important strategy in medical research. It offers several advantages: existing safety data and known side effects, lower development costs and faster implementation, established manufacturing processes. Some successful examples of repurposed drugs in cancer treatment include thalidomide, originally a sedative, now used for multiple myeloma; metformin, a diabetes drug showing promise in cancer prevention; aspirin, being studied for its potential to reduce colorectal cancer risk. This approach has led researchers to investigate numerous existing medications, including antiparasitic drugs like Ivermectin and Fenbendazole. What is Ivermectin? Ivermectin was discovered in the 1970s and won its developers the Nobel Prize in Medicine in 2015. It's FDA approved for treating certain parasitic infections in humans and is on the WHO's list of essential medicines. The laboratory evidence for ivermectin's potential anti-cancer properties is intriguing. Studies have shown it can inhibit the growth of various cancer cell lines, including breast cancer, colorectal cancer, and leukemia cells. Research published in journals like Nature Communications has demonstrated that ivermectin may selectively target cancer stem-like cells, the cells often responsible for treatment resistance and recurrence. Scientists have identified several possible mechanisms of action: inhibition of the PAC1 ACT signaling pathway, blocking the WNT-TCF pathway, inducing immunogenic cell death, potentially making tumor cells more vulnerable to other treatments. Moving from laboratory to clinical evidence, a Phase 1 trial at Johns Hopkins explored combining ivermectin with temozolomide for patients with newly diagnosed glioblastoma. While primarily focused on safety, the combination proved tolerable at certain dosages. Epidemiological studies have noted correlations between regions with widespread ivermectin use for parasitic diseases and lower cancer rates. However, correlation doesn't equal causation, and many other factors could explain these differences. As of late 2024, several clinical trials were investigating ivermectin's potential in different cancer types, including a Phase 2 trial combining ivermectin with chemotherapy for colorectal cancer, a study exploring its combination with immunotherapy for melanoma, research examining its effects on the tumor microenvironment in breast cancer. Important limitations to consider: most evidence remains preclinical. Optimal dosing for potential anti-cancer effects is unknown. Long-term safety at higher doses hasn't been established. Potential interactions with standard cancer treatments require further study. What is Fenbendazole? Fenbendazole is an anthelmintic medication used primarily in veterinary medicine to treat parasitic worm infections in animals. Unlike ivermectin, it is not FDA approved for human use. Laboratory studies have shown several interesting findings. A 2018 study in Nature Scientific Reports demonstrated that fenbendazole could reduce tumor growth in non-small cell lung cancer xenograft mouse models. Research suggests it works by disrupting microtubule dynamics, which are essential for cell division, inducing mitochondrial dysfunction in cancer cells, creating oxidative stress that may preferentially affect cancer cells, potentially interfering with glucose uptake, essentially starving cancer cells. Laboratory testing shows activity against multiple cancer cell lines, including lung cancer, colorectal cancer, lymphoma, prostate cancer. Case reports and anecdotal evidence. The most widely discussed case involves Joe Tippins, who claimed his advanced small-cell lung cancer went into remission after taking fenbendazole, marketed for deworming dogs, along with other supplements and some conventional treatments. His story gained significant attention online and led to what's now known as the Joe Tippins Protocol. However, it's crucial to note, this represents a single anecdotal case without controlled conditions. Multiple treatments were used simultaneously. The outcome lacks formal medical documentation in peer-reviewed literature. A small collection of other case reports exists, primarily from integrative medicine practitioners, describing patients who self-administered fenbendazole. Some reported disease stabilization, but these reports lack standardized assessment criteria, control groups, peer review, long-term follow-up, research limitations. The limitations for fenbendazole research are even more significant than for ivermectin. Far fewer formal clinical trials are underway due to its veterinary-only approval status. Safety data for human use is limited, particularly at doses that might be required for potential anti-cancer effects. Pharmaceutical-grade formulations for human use don't exist, raising quality control concerns, self-administration risks, unknown drug interactions with conventional cancer treatments. One claim sometimes associated with these medications is that cancer is closely related to parasites. Let's examine this statement carefully. There are some documented connections between certain parasitic infections and increased cancer risk. Schistosomiasis is associated with increased bladder cancer risk. Liver flukes are linked to cholangiocarcinoma. Some parasitic infections create chronic inflammation, which can contribute to cancer development. However, the scientific consensus does not support the claim that parasites are a primary cause of most cancers. Cancer development is understood to involve genetic mutations, environmental factors, lifestyle influences, age-related cellular changes, immune system dysfunction. What's more likely is that these antiparasitic medications have mechanisms of action that coincidentally affect cancer cells through pathways unrelated to their antiparasitic effects. To put this research in context, let's briefly review how potential cancer treatments typically progress. Laboratory studies. Initial testing in cancer cell lines and animal models to establish potential efficacy and basic mechanisms. Preclinical development. More rigorous testing to determine safety profiles, optimal dosing, and potential drug interactions. Phase 1 clinical trials. Small studies primarily focused on safety and determining appropriate dosages in humans. Phase 2 clinical trials. Larger studies examine if the treatment shows efficacy against specific cancer types. Phase 3 clinical trials. Large-scale randomized controlled trials comparing the new treatment to standard care. Regulatory review. Evaluation by agencies like the FDA to determine if benefits outweigh risks. Post-approval monitoring. Ongoing assessment of real-world effectiveness and side effects. Currently, ivermectin has entered early clinical trials for cancer, while fenbendazole remains primarily in the laboratory study phase for cancer applications. This area of research raises several important considerations. For patients, self-medication with these drugs for cancer carries unknown risks, potential drug interactions with conventional treatments, quality control issues with veterinary formulations. Delaying proven treatments could allow disease progression. For researchers, ethical responsibility to follow evidence without bias. Importance of rigorous clinical trials. Need for transparent reporting of both positive and negative results. Balance between innovation and patient safety. For healthcare providers, staying informed about emerging research. Communicating honestly with patients about evidence limitations. Supporting patients' interest in complementary approaches while emphasizing evidence-based care, reporting observed outcomes to contribute to the knowledge base. So where does this leave us? The investigation of antiparasitic drugs like ivermectin and fenbendazole for cancer treatment represents an intriguing area of research with promising laboratory findings, plausible biological mechanisms, some preliminary clinical investigations, compelling anecdotal reports. However, neither medication is currently approved for cancer treatment, and the evidence remains insufficient to recommend their use outside of clinical trials. For those interested in this research, follow updates from clinical trials registered on clinicaltrials.gov. Look for peer-reviewed publications in reputable medical journals. Discuss emerging research with qualified healthcare providers. Consider participation in clinical trials when available. Science advances through methodical investigation, and today's laboratory findings may indeed lead to tomorrow's treatments. The exploration of repurposed medications like Ivermectin and Fenbendazole deserves continued research attention and funding. I hope this video has helped clarify where the evidence currently stands. If you found this information valuable, please like and subscribe for more evidence-based discussions on emerging cancer research. Thank you for watching.